Hi, I'm Alison Crammond, and this presentation will explore the use of recombinant proteins in medicine, and will be presented by myself, Emma Gosper, and Justine Purdy. I will begin by giving a brief introduction into recombinant proteins and their purposes, before moving on to discuss the history and development of the technique. Emma will then discuss how the technique works and its current applications, before Justine moves on to discuss the advantages, disadvantages, and potential future applications of the technique. So what are recombinant proteins? A recombinant protein is a type of modified protein whose code is encoded by a recombinant DNA. A recombinant DNA is a DNA artificially created from two or more sources, which have been covalently linked to form new combinations. This involves the isolation and cloning of a DNA fragment encoding for a specific protein into an expression vector, which is a plasmid or virus designed for gene expression in cells. The protein is then expressed in protein expression systems such as bacteria, yeast, insect, or mammalian cells. Bacterial cells are the most commonly used as they are easy, efficient, and inexpensive to culture. In fact, E. coli was the first organism used in recombinant DNA technology and is still the most commonly host cell used today. What are recombinant proteins used for? Recombinant proteins have a wide spectrum of applications in treating diseases and improvement of human health. This includes diagnosis, treatment, and prevention of various diseases in humans. For example, they can be used in pharmaceuticals, vaccines, hormone therapy, and antibodies. Here are some specific examples of recombinant proteins and their uses in medicine, and the disease or diseases they can be used to treat. Coagulant factors, for example, coagulant factor 8, can be used in the treatment of haemophilia A and B by promoting blood clot formation. DNase 1 is used in the treatment of cystic fibrosis. Erythropoietin can be used to treat anemia by promoting red blood cell formation. Insulin is used in the treatment of diabetes. Alpha interferon can be used to treat some leukemias and other cancers, as well as hepatitis B and C. Beta interferon is used in the treatment of multiple sclerosis. Interleukin 2 is used in immunotherapy of cancer. Tissue-type plasminogen activator can be used to treat acute myocardial infarction. Monoclonal antibodies can be used to treat lupus and rheumatoid arthritis. Somatotropin is used in the treatment of growth hormone deficiency. And finally, vaccines, for example, hepatitis A and B. And these are just some examples of the types of recombinant proteins and their uses. There are many, many more types and many more uses in medicine. I will now discuss the history and development of recombinant proteins and recombinant DNA technology. During 1969 to 1970, Paul Berg and Peter Loban devised a way of creating recombinant DNAs in vitro to be used to manipulate genes across species. In 1971, Douglas Berg and colleagues isolate the first plasmid bacterial cloning vector. During 1971 to 1972, David Jackson and colleagues, along with Peter Loban and A.G. Kaiser, develop a method for joining DNAs in vitro. In 1972, Loban and Kaiser published a hypothesis in the Journal of Molecular Biology, which described the method of separating and amplifying genes and inserting them into a new host cell. In that same year, Jackson and colleagues create the first recombinant DNA in vitro. However, further research is delayed by some leading researchers pending further investigation and understanding of potential biohazards, such as the cancer-causing potential of laboratory-altered viruses. During 1972 to 1973, several conferences are held to discuss the regulation and safe use of recombinant DNA technology. During that same period, Stanley Cohen and colleagues isolate a new cloning vector and create bacterial intra- and interspecies. In 1973, John Marrow and colleagues clone and propagate ribosomal DNA genes from a eukaryote in E. coli. Researchers concerned about potential biohazards request that the National Academy of Science establish a committee to examine risks and benefits of recombinant DNA research and propose guidelines for such research. In 1974, the chair of the National Academy of Science presented their recommendations. These included the deferral of risky recombinant DNA research and the establishment by the National Institute of Health of an advisory committee to assess the risks involved and develop procedures to limit such risks. In that same year, the National Institute of Health creates the Recombinant DNA Molecule Program Advisory Committee. In 1976, the National Institute of Health releases the first guidelines to be used in recombinant DNA research. In 1977, Genentech, the first genetically engineering company founded by Robert Swan and Herbert Boyer, used genetically engineered bacteria to synthesize somatostatin, a human growth hormone. This marks the first recombinant expression in E. coli. In 1978, Boyer produces a synthetic version of human insulin. In 1980, Cohen and Boyer receive US patents for gene cloning. And in 1982, the US Food and Drug Administration approves the first recombinant protein for clinical use, and this was insulin. During 1986, the FDA approves 3 new recombinant proteins for use in clinical practice. These were the first interferon drug to treat cancer, the first recombinant vaccine to prevent hepatitis B, the first recombinant tissue plasminogen activator to treat myocardial infarction. In 1987, yeast chromosomes are invented by Maynard Olson and colleagues, and these are expression vectors for large proteins. In 1989, erythropoietin is approved for treatment of anemia in chronic renal disease. In 1992, the first recombinant coagulant factor is approved to treat haemophilia. In 1993, the first recombinant protein to treat multiple sclerosis is approved and is the first therapy to be effective in altering the natural history of relapsing and remitting multiple sclerosis. In 1994, recombinant DNase 1 is approved for treatment of cystic fibrosis. In 1995, the first vaccine for hepatitis A is approved. 1997 saw more recombinant proteins approved for clinical practice than in the previous several years combined. This included treatments for AIDS, some cancers, hereditary disorders, human growth hormone deficiency, as well as vaccines for hepatitis B and C. By the first decade of the 21st century, hundreds of proteins were being produced recombinantly for use in medicine, and about 50 had been approved for clinical use. Today, many recombinant proteins are part of standard therapy, and many have been used in clinical practice for over 20 years, and are considered safe, highly effective, and absent of side effects.
Hi, my name is Emma Gosper and I will be carrying on from Alison. I will be covering how does recombinant protein work and the current applications of recombinant proteins in medicine. So how does it work? Producing recombinant proteins involves DNA cloning, protein amplification and protein purification. The polymerase chain reaction can also be used. The first step to producing recombinant proteins is to perform DNA cloning. In DNA cloning, the first step is to identify and obtain the DNA segment to be cloned. The DNA fragment is obtained by cleaving the chromosome with restriction endonuclease. The second step is to select DNA capable of autonomous replication. Once chosen, the cloning vector is cleaved with restriction endonuclease. The third step is to join the 2 fragments covalently. DNA ligase is used to link the cloning vector to the DNA fragment to be cloned. The fourth step is to move the recombinant DNA from the test tube to a host organism, so that the DNA is introduced into the host cell so that many copies of recombinant DNA can be completed. This is performed by either using transformation, which involves incubating the cells and DNA together, and providing a heat shock, which causes the cells to take up the DNA, or by using electroporation, which involves a high voltage pulse to render the bacterial membrane permeable. Step 5 involves identifying the host cells that contain the recombinant DNA. This is because not all cells will take up the recombinant DNA. In order to do this, the cloning vector could have a selectable marker added which will allow the host cells to survive and the lacking cells to die. For example, antibiotic resistance gene. A screenable marker can be used which is a gene encoding a protein that causes the cell to produce a fluorescent colour. The bacteria are then spread onto an agar plate which contains an antibiotic and incubated overnight. The only bacterial cells to have successfully transformed will contain the plasmid and the antibiotic resistance gene. All of these steps described can be seen in this image to the right. The recombinant protein then needs to be amplified. Proteins are produced in very high amounts for commercial use. Different systems are used for this. Bacteria. This is the most common host for protein expression, especially E. coli. They are well understood and are grown in fermenters and produce large amounts of commercial protein. Yeast. The principle for yeast is similar to using bacteria, however the cloned genes must be linked to promoters that direct high-level expression in yeast cells. Insects and insect viruses. Baculoviruses are insect viruses that infect larval hosts, act as parasites, kill the larvae, and turn them into factories for virus production. Late in the process, the viruses produce large amounts of proteins. The genes for these proteins can be replaced with a gene of interest and produced at very high levels. Mammalian cells in culture. The cloned genes are introduced using a virus. This takes advantage of the natural ability for a virus to insert its DNA into a cell. There is an image of this to the right here. The cloned protein is then expressed. Growing mammalian cells in tissue culture is expensive, so this method is often only used to test the function of a protein in vivo. The recombinant protein then needs to be purified. This is vital for the function, structure, and interactions of proteins and involves the separation of the protein of interest from all impurities. This can be performed using affinity chromatography. This is one of the most efficient methods which uses the reversible interactions between a protein and its ligand. However, many proteins do not bind a ligand that can be immobilized on a column matrix, so a tag is needed. The gene for most proteins can be altered to express a fusion protein, which is the tag, which can then be purified by affinity chromatography. Ion exchange. This uses the reversible interactions between a protein and a charged chromatography medium. Size exclusion, which separates protein based on size. Hydrophobic interaction, which uses the reversible interactions between a protein and a hydrophobic ligand bound to the chromatography matrix. The polymerase chain reaction, or PCR, can also be used to produce recombinant proteins. PCR can be used for the replication of specific DNA or RNA sequences and allows for increased protein expression through codon optimization and sequence alterations. The steps involved include denaturation, the separation of two nucleotide strands of a DNA molecule, primer annealing, the primers bind to single-stranded DNA, elongation, nucleotides are added to the primers in the 5:3 direction to form a double-stranded copy of the target DNA. Each cycle only takes a few minutes, so repeated cycles can produce very large amounts. I will now discuss the current applications of recombinant proteins in medicine. I will cover the broad categories and then discuss 3 specific examples. Currently, there are a number of different therapeutic recombinant proteins available. These include recombinant antibodies. These are protein molecules that recognize and attack foreign bacteria and viruses in the body. Recombinant enzymes. These speed up metabolic reactions, for example, DNase for cystic fibrosis. Recombinant hormones. These function as a communication between different organs and tissues, for example, insulin for treating diabetes. Recombinant cytokines. These are involved in the innate and adaptive immune system. Recombinant chemokines. These are signaling proteins secreted by cells. Some are pro-inflammatory and some are homeostatic. Recombinant neurotrophins. These promote the growth and survival of neurons. These provide the therapy for disorders including neuronal degeneration and physical trauma, involving the severing of nerve connections. Recombinant growth factors. These stimulate the growth of cells, for example, erythropoietin for treating severe anemia with kidney damage. One example of a recombinant protein is insulin. Insulin is a pancreatic hormone that regulates the cellular uptake, utilization and storage of glucose, amino acids and fatty acids, and inhibits the breakdown of glycogen, protein and fat. Recombinant human insulin is identical in function and structure to the native human hormone. Recombinant insulin is used therapeutically to treat high blood glucose, including diabetes type 1 and 2, gestational diabetes, diabetic ketoacidosis, and acute stroke. It is produced by inserting the human insulin gene into the E. coli bacteria. The bacteria are fermented and purified, which produces insulin for human use. It is typically administered by an injection under the skin. A second example is recombinant human growth hormone, or HGH. HGH is required for normal human growth and development. It stimulates the expression of insulin-like growth factor 1, increases calcium retention, bone mineralization, and muscle mass, promotes lipolysis, and reduces the uptake of glucose. Recombinant human growth hormone has therapeutic applications for the treatment of dwarfism, bone fractures, skin burns, bleeding ulcers, and autoimmune deficiency syndrome. Recombinant HGH treats growth hormone deficient children to reach final height and to increase insulin-like growth factor, osteocalcin, and bone density. Provides therapy for growth hormone deficient adults and is used to accelerate wound healing. Recombinant HGH is administered via injection. Here is a high-level flowchart showing how recombinant HGH is produced, using E. coli as the host strain. A third example is coagulation factor VIII. Coagulation factor VIII is made in the liver. It circulates in the bloodstream bound to another molecule called the von Willebrand factor until an injury occurs. Factor VIII is then activated and separates from the von Willebrand factor with another coagulation factor called factor IX. A chain of reactions are then set off in order to form a blood clot. Haemophilia is caused by the failure to produce the proteins required for blood clotting, factor VIII for haemophilia A or factor IX for haemophilia B. The altered or missing protein cannot participate effectively in the blood clotting process. As a result, blood clots cannot form in response to injury, which leads to excessive bleeding. Treatment involves intravenous infusion of recombinant factor VIII, concentrate immediately or within 1 hour of the onset of bleeding. Prevention involves regular prophylactic infusions.
Hi, this is Justine Perdue and I'll be talking about the advantages, disadvantages and future applications of recombinant proteins in medicine. Recombinant protein technology is vitally important in medicine via diagnostic or therapeutic applications. Advances in biotechnology continually improve the purity and safety while increasing the availability of proteins that were previously isolated from human or animal sources. Source safety. Proteins derived from a native source may be potentially dangerous, harbor pathogens, or even be a pathogenic species in itself. Recombinant protein production in a non-pathogenic, non-toxic host circumvents such potential difficulties. Source availability. Many proteins are produced naturally at very low concentrations, with high-scale production limited by origin availability. Recombinant protein technologies overcome this issue, delivering a product that's consistent in quality while minimizing side effects. Some previously mentioned examples were improved upon with the development of recombinant proteins. Insulin was initially derived from animals. However, animal insulin is different from human insulin. It cannot be purely isolated, often causing immune responses and allergic reactions. The predominant supply of insulin currently in use is produced by recombinant proteins synthesized in E. coli or yeast cells, eliminating the risk of potentially fatal reactions. Growth hormone is species-specific, which renders it critical to use human growth hormone in human medicine. Until the mid-80s, the only source was harvested from pituitary glands of human cadavers. In 1985, the transmission of Creutzfeldt-Jakob prion disease was associated with contaminated cadaver preparations. Recombinant protein versions of human growth hormone not only overcame problems of source availability, but also assured a much safer product. Coagulation factor VIII was originally derived from concentrated forms of donated plasma. However, contaminated sources led to transmission of blood diseases in the 80s, with some companies continuing to sell contaminated factor VIII even after new heat-treated versions were available. Since the 90s, recombinant factor VIII production has overcome source availability issues, but most importantly, is in the prevention of disease transmission during replacement therapy. The creation of novel proteins are thanks in advances to molecular biology. They facilitate modifications ranging from single amino acid deletions or insertions, larger fragment substitutions, and even the creation of chimera proteins with the fusion of protein sequences combining two paternal proteins. This bioengineering improves recombinant protein stability, solubility, or specificity in therapeutic applications. While recombinant proteins are highly beneficial, there are some shortcomings in the technology. Post-translation modifications. Bacterial systems are predominantly used because they are high expressors, easy to scale up at a relatively low cost. Many human proteins require the post-translational modification of glycosylation, yet bacteria does not allow for this, so eukaryotic cells must be used. Glycosylation in yeast cells is still vastly different from that produced in humans, resulting in incorrectly processed and partially unfolded proteins, rendering them biologically inactive. Protein production in high eukaryotic cells is preferred, but this process is unfortunately more expensive with a much lower expression rate. Antibody-mediated reactions of varying degrees occur with recombinant proteins and may interfere with treatment efficacy. Serious complications can occur when haemophiliacs develop inhibitors to injected factor VIII, resulting in a tolerance to the clotting factor. Patients treated with interferon alpha and beta interferons also show evidence of inhibitors, thus decreasing therapeutic effects of subsequent treatments. The future of recombinant proteins in medicine is in improving current systems and potentially shifting those expression systems in situ. Piggyback transplant technology. Conventional gene transfer methods rely on random plasmid integration, resulting in variable expression levels. Large cell numbers often produce insufficient yields due to few high-expressing clones. This newer system enables genes of interest in the vector to be moved from the original site and integrated into a specific chromosomal site. It is effectively a cut-and-paste mechanism between vector and chromosome. While this is a more efficient and effective alternative, it is still currently only for research applications. Ribosome engineering. Recombinant protein production has evolved through the synthesis of biopharmaceuticals to improve efficiency. There are now very early discussions for efforts to redesign and reshape parts of the ribosome itself, potentially introducing new functions and new proteins. Finite sets of protein monomers exist naturally, which then produce a limited amount of biopolymers. By expanding upon Mother Nature's library of ribosomal amino acids, this could generate new classes of enzymes and future therapeutics. A DNA vaccine is when a plasmid containing a DNA sequence encoding for specific antigens is directly injected into recipient cells. Once processed and displayed on the surface of the host cell, the immune system is alerted, triggering an immune response. The potential advantages over current techniques include both B and T cell responses, an improved vaccine stability, and the absence of any actual infectious agent. These DNA vaccines would directly introduce the antigen, leading to a more immediate and permanent immunity. Animal immune responses have been observed using infectious agents. However, no DNA vaccines have been approved for human use and is still in the clinical testing stages. Current research demonstrates that the human microbiome has significant implications in health and disease. The next generation therapies are recombinant probiotics. Specific modifications of living cells will allow recombinant expression of therapeutic biomolecules. These microbes will be engineered to sense and remedy disease via the production of therapeutic bacteria in situ, which will then assemble to counter a pathogenic invasion or undesirable microbiota. Microbiome-mediated homeostasis has great potential for decreasing risks of infection, cancer, metabolic, and immune disorders. However, microbiome engineering at this level exists currently only in theory.